our YouTube channel, NCERT official. You can also write your questions, your queries in the live chat box of our YouTube channel. You're watching us live on SWAYAM Prabha's channel number 31, Kishore Manch. Let's meet our expert for this session, who is going to give us information on this topic. So we have with us Mrs. Reena Mohapatra. Welcome, ma'am.
Thank you, Tanvi.
Ma'am is from DAV Schools, Bhubaneswar, and she is going to clarify my doubts as well. So let's begin with this discussion. Ma'am, of course, this is a biology session, so there can't be only theory. There will be some practical as well, which I'm sure you're going to show us something. So how are you going to begin this session?
And good afternoon, all the viewers. So today our session, as already it has been told, sexual reproduction in flowering plants. So in this we are getting three terms. One is reproduction, one is sexual, and another is flowering plants. Reproduction is the one of the most, I won't say that this is the most important, but it is one of the most important characteristic of living organisms.
Okay.
Sexual reproduction, there are different types of reproduction, and sexual reproduction is regarded as most advanced type of reproduction. As you know, this also provides varieties to the organisms and increases its chances of survival. Then next comes flowering plants.
Okay.
The group of flowering plants, otherwise called as angiosperms. Around us nowadays, which plants we see, almost 95% of them are angiosperms. So these plants have developed flowers as well as fruits. As over previous groups.
Okay.
In previous groups, these two characteristics were not there, flowers and fruits were not there. So these flowers have been developed for sexual reproduction, and this has been already taught in previous class, standard 11. So all of the students, they know about it. Just for recapitulation, I told about it. Okay. Now, before starting our session, I'll I would like to show you some on the slides. Please, you see here, I think you can make out that towards the left we have we are having say there are bryophytes, that is mosses, as well as here you find pteridophytes are there. Bryophytes have their reproductive structures as like the small stalk-like structures, whereas pteridophytes have spores on the underside of their leaves. As you can find here, this is a pteridophytic leaf, and you can find here that these are the spores present here. The spore-bearing structures are called as sporangia. They are released during the drier season of the month, and they spread on the ground and they give rise to new plants.
Okay.
Let's see this next one. Here you find that gymnospermic cones are there. In the upper left corner you find there are cycas cones, and lower left corner you find there are pinus cones. Here I'll show you some. This is another pinus cone. You can make out it's looking similar.
Yeah.
So here you find these cones are, how do they look? They look woody structures.
Yes, ma'am.
So I've shown you in three groups what is the condition. Here also spore-bearing structure is present in gymnosperms in this cone. In pteridophyte it is present on the underside of the leaf, and in case of moss, the spore-bearing structures are borne by a small stalked capsules. Okay, but still they are reproducing. So my question is here, if here flower is not there but still reproduction is taking place, so what was the need of flower in angiospermic plants? In angiospermic plants, flowers had been developed.
Okay.
So is the flower really necessary for sexual reproduction? Let's discuss now.
Sure.
Now look here, here in this picture you can find there are these flowers are very colorful and quite large, which can be seen with naked eyes nicely. But all flowers are not like that. That they are small, they are not so colorful. Different types of plants wear different types of colors. So our question was, is the flower very much necessary for sexual reproduction? So if we see here, we'll say yes, it is. Here I'll take you one flower and show you. See, by now you know in standard 11 you have learned that different parts of a flower has got calycula, androecium and gynoecium. Outer parts of the flower, they protect the inner parts of the flower. Okay, androecium and gynoecium are protected. Apart from that, you can find flowers are so showy, so large and attractive that they can attract pollinators. And so when they attract pollinators, this will help in attracting different types of pollinators and help again in better pollination and better chance of survival. So for all these reasons, certainly it's in angiosperms, as flowers had been developed, so today angiosperms are dominating on the earth's surface. Their number is maximum. So flower is a necessary factor for sexual reproduction. It is already proven. Okay.
Okay.
Now the question is, what causes flowering?
Flowering.
To what flowering will be there? Okay. Now see, there are certain conditions which say in almost all the plants you'll find from seed. When a plant is growing, this plant will not bear flower. That when a young plant comes out from the seed, you'll never find flower in this. So a plant grows from a seed, then it will go through a certain stage. This stage is called as juvenile stage. From juvenile stage, gradually they'll move towards adult stage. But in this adult stage is also divided into two phases. What is that? Adult non-flowering phase and then this non-flowering adult phase will proceed towards adult flowering phase. That is, from the beginning there is no flower in it. Gradually, as the plant becomes mature, becomes adult and it reaches flowering phase, then it will bear flowers. So the question comes, what causes flowering? When there was no flower and in bearing of the flower, what are the conditions? There are two conditions. Internal factors are there, external factors are also there. Among external factors you find there is photoperiodism is the main factor. I'm not going into details because already in the previous year in standard 11 you have learnt about it. And internal factors are several hormones like gibberellin hormone, etc. is there. Apart from that, you also find among external factors, if stress is there due to lack of food, low temperature or high temperature, or there is a lack of water, then also flowering will be inhibited. So these are some of the factors for flowering. Next is we see, okay, flowering, we find there is flowering conditions have been availed, the external conditions and internal conditions have been fulfilled. Now flowering should be started. But from where flower will come? In a young plant we don't find any specific part where flowering is there. You know that the plant is growing, as you find in this case, this is the vegetative apex. It grows and it gives rise to leaves and it increases the height of the plant. Okay. So when it has been longitudinally cut and observed under the microscope, this is the anatomical structure of this vegetative apex. Now in this case you can find this dome-shaped structure is present here, which shows it is the tip of the stem, whereas these are the leaf primordia. These are the leaf primordia. This leaf primordia will divide and redivide, and they'll give rise to leaves. So here we don't find any place where a flower will be grown. So what's happening when all the conditions for flowering has been achieved? How flower will be born? Okay. Now what happens when these conditions have been achieved? Then you'll find that these, say particularly gibberellin hormone, will reach up to this tip of the apical meristem when the plant decides to bear flower. So now in this case, what would happen? Certain changes will take place in the tip of the plant. What are the changes? Normally you find that a stem has got nodes and internodes. So these nodes and internodes are quite long, and you find from the nodes leaves are coming out. Okay. When a plant decides to have flowers, these internodes will become shortest. As a result, what would happen? All the nodes will come over each other. Due to absence of internodes, the nodes will come over each other.
Okay.
And then the leaves, which leaves were supposed to come out from the nodes, they come out from same place. Okay.
Okay.
And these leaves, each of these leaves will be modified to form parts, different parts of a flower, that is calyx, corolla, androecium and gynoecium. Followed?
Yes, ma'am.
This is of course a completely different topic. So it's a long process, it takes place. Okay.
Okay.
So in brief, what can we say? This each and every part of a flower, what you see that here, which parts you find here, that is calyx, corolla, say, and androecium and gynoecium, all these parts are nothing but modified leaf. So in short, we can say flower is a heterophylla structure. Flower is a heterophylla structure. What does it mean? That flower has got leaves in different forms and shapes. Okay. What are these leaves? These are the parts of a flower. Okay. So we have got it. So now what we see that vegetative apex has been converted to a reproductive apex, which is bearing flowers. So now let's come to the flower. Here all of you know that a flower has got 4 different parts: calyx, corolla, androecium and gynoecium. Calyx and corolla, they are associating parts and they help in reproduction. We'll concentrate our attention on androecium and gynoecium. Androecium is the male reproductive part. As I had shown you in this flower, here these are the stamens. In different flowers, number of stamens, shape, size and forms of stamens vary. In this flower you can find, I have removed some, otherwise here 5 stamens were there, and which were also attached with the petals. Okay.
Okay.
So here you find all the stamens in a flower, together they constitute androecium. So androecium constitutes stamens of a flower.
Okay.
Understand? So now let's see part of a stamen. So here you can see here these are the stamens. Okay. So each stamen has got 2 parts. This is the filament and this is the anther. Okay. In most of the angiospermic plants, again we find each anther has got 2 lobes. You might have seen here one lobe is present. Just I'll remove it and show you. Here this side one anther lobe is present, here another anther lobe is present.
Okay.
So most of the stamens have got 2 anther lobes. Again, you can find here each anther lobe possesses another longitudinal, two longitudinal structures, which are called as theca. So this side two such structures are present, and here also two such structures are present. As you find here you can find this is the filament, and this whole thing is the anther. Each anther has got, this is one anther lobe, and the other side, that is another anther lobe. Two anther lobes are fused by a sterile mass of tissue which is called as connective. Each anther lobe again has got two longitudinal lobe-like structure. This is one and this is one. So these two are called as theca. This is a theca, this is a theca. So in each anther lobe, two theca are present. Such kind of anther is called as dithecous anther, as there are two theca per anther lobe. Okay. So as there are two theca per anther lobe, and there are two anther lobes per anther, so how many theca will be present in a in an anther? Certainly 4.
Yeah.
Okay. So 4 theca are present in each anther, but that is not the always that is not the case. You can take, this is China rose. In case of China rose, you'll find that each anther possesses two anther lobes, but each anther lobe possesses a single theca. That's why China rose is said to be having monothecous anther. Okay.
Okay.
So now let's go to the next slide. So I think this is clearly visible to you.
Yes, ma'am.
Now you can find that here I've taken.
Ma'am, the title says TS of anther. What does
Yes.
TS stands for?
TS stands for transverse section of anther.
Okay.
That is the basic, section in biology. So this is the, as I had shown you in this picture.
Yeah.
You had found that this was one anther lobe. This is another anther lobe, and these are two theca of one anther lobe, and this one, and at the backside another one is there. Two theca are present per anther lobe. You can find in between two theca of the same anther lobe, a depression is usually present. This depression quite often, it shows the site of dehiscence. Through this part, the anther dehisces and releases mature pollen grains. Okay, so when we take transverse section of this anther, how does it look? Let's see. When we take transverse section of the anther, it will look like this. So 4 theca per anther are present. 1, 2, 3, 4. 4 theca are present.
Okay.
These theca are longitudinally arranged. And inside each theca, you can find a round or oval mass of tissue is present. So this region, this sac-like structure, which consists this mass of tissue, is called as sporangium. And as here we are discussing about male part of the plant, so this sporangium will be called as microsporangium. Micro itself means small, but micro term is also associated with sexual parts of many male reproductive parts.
Okay. So, ma'am, had this been the female part, the name would have been different?
Mega, yes, it would have been called as mega or macro.
Okay.
Sporangium. Okay, so this is microsporangium. As there are 4 theca per theca, 1 sporangium, microsporangium. So how many microsporangia will be present? Certainly 4 microsporangia will be present in each anther.
Yeah.
Okay. Now, how can we see it? Now see, here we have tried this anther I've taken. This is a datura flower. All of you can see this is a datura flower. Datura flower has got quite long anther, so it is easier to take. You can do it at home also. You take a transverse section, a thin section you take, and then you keep it on a drop of water and in a glass plate or something, so that light will pass through it, and then you can see here. Can you see here?
Yeah.
This structure here, how does it look? That characteristic butterfly shape, can you find?
Yes, ma'am. The light pink butterfly.
Butterfly shape, that is the upper portion of it. Does it look like this?
It does, yes.
I'm not telling about color. Shape wise?
The shape, yes, it does.
Okay.
In the center there is a red dot too.
Yes, very, very nice. So this red dot is what? That is connective. This connective is joining two anther-lobes together.
So that's the function of the connective.
Yes, that is the connective holds the two anther-lobes together. Otherwise, it doesn't take part in reproduction. Apart from that, at the red part, you also find some vascular tissues are present. Through vascular tissues, food, nutrients, etc. are passed. Okay, so this is clear. This one, I think here.
Yes, so ma'am. This is very much clear.
Okay, so now let's come to this. Now we'll concentrate on each of the Microsporangium.
Okay.
Okay. So here we'll magnify it, and this is one Microsporangium, a part of one Microsporangium. So you can find this part is called as Microsporangium. This one is Microsporangium. But surrounding Microsporangium, some tissues are present. These are the tissues.
Yes, ma'am.
These are the cells which are arranged in layers. So first, let's learn about it. So now let's see how tiers of anther exactly looks like. Now each part we are concentrating, and here we find from outside to inside. If we move, we'll find the outermost layer which is compactly arranged and made up of comparatively larger size cells, and on maturation of the anther they become thick wall too. This is called as epidermis. Certainly function of epidermis is to provide protection, so they provide protection. Next to epidermis, this layer is called as endothecium. This is also single layered, and same function it performs. It also provides protection. Next to endothecium lies middle layers. Middle layers may be 2 to 3 layers. So here only 2 layers have been shown, so it may be 2 to 3 layers. This middle layers together, main function is same, protection. So these 3 layers provide protection. Next to that you find another layer is present. Yes, another thing I would like to tell you, these 3 layers are continuous. You can say they are continuous throughout the anther.
Right.
Okay. And then comes the 4th layer, which is called as tapetum. But tapetum is restricted to each Microsporangium, that is surrounding each Microsporangium, its own tapetal layer is present.
Okay.
So this is the tapetum.
Ma'am, when you said a middle layer, it's different. It could be 2 or it could be 3. So it depends on the different flowers, or the female or male part.
No, this is here we are discussing only male part.
Right.
Okay. So in female completely it's different, that we'll discuss later.
Okay.
So, but it depends on species to species, where it will be 2 layered, where it will be 3 layered. Now this tapetum layer, which is present, it surrounds the Microsporangium. You can find here the cells are much larger, and these cells look different from the rest of the layers. These tapetal layer cells are large in size, they contain dense cytoplasm, and they contain binucleate to multinucleate condition. They contain the binucleate to multinucleate condition. Now there are many different ways by which this binucleate or multinucleate condition has been achieved. So here a question just I'm putting here. If you get the answer, we'll discuss, otherwise I'll tell you the answer.
Okay.
Now what is the reason that it is having dense cytoplasm and bi to multinucleate condition? How it helps in its function. Okay. And it encloses a mass of tissue internally. You can find here internally, here this tapetum, it encloses a mass of tissue. Here you can find this mass of tissue, which is not fully visible here. Okay. This mass of tissue is, these tissues are called as microsporogenous tissues.
Okay.
Till now we have discussed about epidermis, endothelium, middle layers, tapetum layer, and microsporogenous tissues. And please remember, all of them are having double set of chromosomes, that is, they're diploid in number. And you know, diploid tissues are also called, in plants, they are also called as sporogenous tissues.
Okay.
Okay. Now, gradually what happens, these cells undergoes maturation, and this microsporogenous tissues will show certain development. What are the developments? Let's see. In a young anther, few microsporogenous tissues are present. As the anther matures, these microsporogenous tissues will start dividing mitotically, so that the number will increase. So what we find, microsporogenous tissues in young anther which are 2n in number, and in young anther they'll undergo mitosis to increase their number. Okay, and a time comes when their number is quite large, and when their number is sufficient, then this microsporogenous tissues will stop dividing mitotically. Okay, and these microsporogenous tissues, so these are microsporogenous tissues. Now these microsporogenous tissues will proceed for meiotic divisions. They divide mitotically when their number is quite large, then they stop mitotic division and they proceed for meiotic division. Okay? So when these microsporogenous tissues are proceeding for meiotic division, then these tissues will no more be called as, microsporogenous tissues, but they will be called as microspore mother cell. Still they are diploid. Microspore mother cells now undergo meiosis. They undergo meiosis. When they undergo meiosis, what would happen? Each microspore mother cell will divide to produce 4 cells. Each of the cell will be called as microspore. What will be the ploidy of each microspore? They'll be haploid. As they have been produced by meiosis, so each of them will be haploid. Okay? So each microspore mother cell will give rise to 4 microspores. Initially, and these 4 microspores will be arranged in a tetrad arrangement. How is the arrangement there? These microspores will be arranged in this manner. 3 we can see from the front and 1 will be behind it. So in tetrad arrangement they will be there and they will remain together. At this condition, when 4 of the microspores are remaining together, they are called as microspore or spore tetrad. Gradually, they will develop their individual cell wall, and when hard cell wall is developing, then they become separated. When they become separated, surrounded by cell wall, then they will be called as microspore. So what is the ploidy of microspore? They'll be haploid in number. So I think this has been understood.
Yes, ma'am.
How microspores are forming. Okay, so we have come up to microspores. Okay, So here we find the anther is maturing, and the microspores, by now microspores have also matured.
Yeah.
Each microspore has become surrounded by cell wall. We'll discuss how at the cell walls, what is the anatomical or biochemical structure of this cell wall. So these microspores are single cell structure. They possess a single nucleus and surrounded by cytoplasm and surrounded by thick wall. Gradually it would happen, this nucleus will divide. Now there will be a mitotic division because already it is haploid, so no more meiosis can take place. So there will be a mitotic division, and by mitotic division 2 nuclei will be formed. The one which is large sized, that will be called as vegetative nucleus. That is called as vegetative nucleus. And the small one is called as generative nucleus. So this was the microspore. Please remember, microspore is always single cell.
Okay.
Whenever it divided and became two-celled, because each of the nucleus will gather some amount of cytoplasm around it. So now we can say they are two-cell structure. Whenever it is transformed into two-cell structure, we call it as micro or male gametophyte. Micro or male gametophyte are otherwise called as pollen grains. So at this condition, pollen grains are ready for dehiscence. So by this time, what has happened? As I had told you that these layers, epidermis, endothelium, middle layers, they were protective layers. So these layers are mostly they are made up of thick wall tissues, but at certain places they possess thin wall tissues too, and this thin walled region is called as stomium. So when anther matures and inside pollen grains have been formed, these through stomium the cells will burst, because stomium is made up of thin wall tissues, so their bursting will take place, and when bursting takes place, these pollen grains will come out. As you can find here, here bursting has taken place, here also bursting has taken place, and the pollen grains will be released.
Okay.
Got it? Yes. After that, what would happen? These pollen grains will be carried from one flower to another, or it may be carried from the anther of one flower to the stigma of the same flower. As you know, this process is called as pollination.
Okay.
So till now what we have done, let us just recapitulate. So we can see here, say we were doing the events of sexual reproduction. The sexual reproduction is a very complex process, and it includes several phases. So all these phases we can broadly divide under 3 events. One is pre-fertilization events, next is fertilization events, and next is post-fertilization events. So here we have done here pre-fertilization events. Okay.
Okay.
So in pre-fertilization events, again it consists of 2 parts. First part is called as gametogenesis. That gametogenesis means formation of gamete. Okay. And next is called as gamete transfer.
Okay.
But here, see, till now I've told you about pollen.
Right.
Have I ever told, we have not discussed about gamete formation. So how can we say gamete is forming here? Isn't it?
Yeah.
we have not discussed, so we'll see it now.
Okay.
Now, what's happening? Let's come to the next.
We have got only 3 minutes left.
That's why I was doing. Okay, here a recapitulation. Let's see.
Okay.
We'll do. So this is again that for a brush up. See microsporogenous tissues. As you see here, they have been by mitosis. They have been divided into. They have increased their number. Then after that microsporogenous tissues, they divide by meiosis to form microspore mother cells. Is it clear? So microspore mother cells divide by meiosis to produce microspore tetrad. Microspore tetrad undergoes separation from each other and they form microspores. Microspores divides by mitosis and what do they form? They form, as I told you, microspores divide by meiosis and they form two cells. One is vegetative cell, one is generative cell. And this vegetative and generative cells together form pollen grain. Is it clear?
Yes, ma'am.
Okay. So now, just little bit I'll tell you here. We didn't discuss about gamete till now. In 60% of angiospermic plants, pollen grains are released in this form. In 2 cell stage, they are released. But there are again 40% of angiospermic plants, there you find this generative cell undergoes another mitotic division to form 2 male gametes. Okay, so in 40% of angiospermic plants, pollen grains release, they are dehisced in the form of 3 cell stage. What are the 3 cells? One is vegetative nucleus, vegetative cell, and rest 2 are
male
gametes. So there are 3 cells are there.
Okay.
Understand?
Yes, ma'am.
But what about this 60% case where they are released in 2 cell stage? In their case, what will happen? Pollination will take place. They will reach to the stigma of father plant and their generative cell will divide from male gametes.
Okay.
Is it clear?
Yes, ma'am.
So now you understood that male gametes are forming, certainly they are forming, they may form in the same mother plant where the anther was there, or they may be formed in the stigma of the other plant. So this process of gamete formation is called as gametogenesis.
Okay.
Okay.
Sure, ma'am.
Okay.
So let's take this discussion further in the second part, shall we?
Okay, okay.
Okay, thank you, ma'am. Thank you for explaining till here, and I am definitely looking forward for the second part as well. Thank you so much for being with us.
Great. Thank you.
Thank you to all the viewers who saw this session. I am sure you all enjoyed this session just the way I did. Thank you so much. And if you have any questions, queries, like I said before, we will keep on giving you the numbers, our contact details, so that you can reach us easily with all your questions, doubts and queries. So ciet.kishoremanch@gmail.com is our email ID. Please feel free to contact us, connect with us anytime you want for any subject query, for any query on any topic. Thank you so much, and take care. Stay at your own houses. Don't go anywhere because the lockdown is still there. Thank you so much. Namaste.